Cool. Bye.
Bye.
Really? It's right, you'll see, it's right there to the left.
I've been here for, and it's just. What?
Oh, sorry, is it here? Anyway, right. That's long. It is like that. I don't know why. Oh no. Yeah. Congratulations.
Oh, I didn't even.
It's very sweet. They didn't know that, and they told me, oh, there was a baby falling down. That's not really actually blood work. Yeah. Yeah, we did. Yes, show your father, no. Haven't sent it to him yet.
Oh. Okay, I definitely will. Yes, let me just make sure.
But I don't get it. Here it looks great.
Let me see. Okay. Let's see. Yeah, it looks okay for us.
And I told her it looks great here. But I mean, it's
okay. It's just very pixelated, but I'm not sure
Let's see. Let me show you a different slide. And
if it's the link.
does this look okay? Does this slide still look?
Let me, I'm gonna take a picture of it. It's, I don't know what's, it's just, is it okay for everybody else? I was just saying the screen looks, for me, it's blurry.
Yeah,
Okay,
same for
my
me.
connection. It's very blurry.
It's weird because the PowerPoint looks okay. Let me try one thing. I think maybe it's just I'm showing a portion of the screen. Does that look better? Let's see. Hold on. Does this look better?
It's about the same. I mean, we can still read for the most part.
Okay.
Yeah.
Not sure why. Like, do the words on this, are they legible?
Yeah, we can read these.
They're just slightly unfocused, but they are legible. We can read it.
Okay. Dr. Sorrells, is that your voice that I hear on this call?
Yeah, I'm here.
Okay. Okay, perfect. And then Dr. Singh, are you also here?
I'm here.
Oh, okay, perfect. Okay, so he's on the call.
Oh, he's gonna be.
Yes. Sec. Okay, yeah, so 1, 2, 4. Perfect.
So everybody else.
Yes.
Yeah, so if the audio at any time sounds bad, someone please speak up, but I think we're good to go.
Yeah, you're coming through great.
Okay.
Okay, so I'll give my introduction. So thank you all for coming to Kim's PhD thesis defense. It's a big day for Kim. I'm sure she's thinking, can't believe I made it here, but we did. But I need to do a few formal things as required by the CNUP. First of all, we definitely need to acknowledge Kim's committee. Becky was here, Laura, Mary, Sean, who is joining from Zoom, and Dr. Itisha Singh, who's our guest examiner from Texas A&M. Thank you all for all your insights, all your help, patience with me when I didn't know what I was doing. It's been a great run, and we really appreciate all your efforts. I also need to acknowledge some special guests, a special guest, Dr. K. Joel Berry, Kim's dad, came from Kettering University. I think it must feel very proud. I think it's your youngest daughter, if I'm not mistaken. Youngest daughter getting her PhD, following in your footsteps somewhat. Dr. Berry got his PhD at CMU, so it's a bit of a homecoming. Mechanical engineering, so I think he mentioned all his kids are engineers except Kim, who decided to go venture into biology and neuroscience. But it wasn't too far because you also did some work in artificial intelligence, so it's kind of a little bit of a connection, right, with your dad. So anyway, this is really, for me, a big day too. It's my last PhD student. And to say that we saved the best for last is probably an understatement, because it's been such a joy to train Kim. I remember when you first came to see me, I was impressed by your insightfulness, your inquisitiveness, and was thinking, like, why would you want to come here? You were so far afield from what you know, what you do, what you've done. But you decided to join, and it's been a great ride. Kim came from Ohio State, where she majored in neuroscience, I guess. Yeah. Mostly did a lot of behavioral work there. Was an award winner there. Since she's been here, she's been funded by, different awards. She's got an award from Enneagram Society, from Farm, from CNUP, from the Provost Office. We're really excited that she got her first author paper, just published literally a couple of weeks ago. So that was a huge undertaking. And I think what I learned about Kim was that not only is she super hardworking, but I think a key feature of being a scientist is being fearless. Saying, hey, this is taking me in a whole different direction that I had no idea I was going in, but it's interesting. I want to know it, and I'm going to just dive in head first. Sometimes it feels like you're diving into a pool with no water, but I tried to spare you from that. Put a little bit of water in the pool, and you did so well. So it's a great honor for me to present my last student. I'm excited to hear the talk again. And I'm looking forward to a really, really bright future. And it's, like I said, been a joy for me. And said I don't want to get too emotional because it is an emotional day for me too. But Kim, I'll leave it for you.
Okay, thanks.
All right, thanks, Don, for that warm and thorough introduction. Thank you to my entire committee for being here. I really appreciate your mentorship. I chose you guys all for your commitment to scientific excellence, as well as your commitment to training the next generation of scientists. And with that, I'll jump straight in. So the title of my talk is Multi-omics Assessment of Glucocorticoid Action in Mouse Neural Stem and Progenitor Cells. But let's first talk about stress. It happens to all of us. I may be even feeling a little bit of it right now, but that's okay. We all have our own ways of dealing with stress through diet, exercise, or obsessing over your pet for a little bit. And these are all great ways to feel a little bit less stressed about the daily tasks that we all juggle. Luckily, thanks to human evolution, our neuroendocrine system developed a biological means of reestablishing physiological homeostasis after experiencing a physical stressor or a psychological stressor. This occurs through activity of the HPA axis. So this is a neuroendocrine system connecting the hypothalamus and the pituitary in the brain to the adrenal cortex, resulting in the coordinated synthesis and release of these glucocorticoids. So these glucocorticoids are going to be our primary stress response hormones that function to kind of bring our body back to baseline levels after experiencing that physiological or that psychological stress. So that main stress hormone in humans is cortisol, whereas in rodents, it's going to be corticosterone. In brief, how this system works is stress causes a release of CRH from the paraventricular nucleus of the hypothalamus, which then stimulates the synthesis and release of ACTH from the pituitary, which again subsequently stimulates the synthesis and release of glucocorticoids from specific cells within the adrenal cortex. These glucocorticoids in turn provide negative feedback to each level of the HPA axis, subsequently terminating its own activation. And we know that this negative feedback mechanism is central for the reestablishment of physiological homeostasis. So if you disrupt this negative feedback mechanism, we know that it's a major pathophysiological contributor to the development of stress-related diseases. And then further regulation of this HPA axis is provided at extra hypothalamic regions, including the hippocampus and the amygdala, as well as the prefrontal cortex. So as major regulators of physiological homeostasis, these glucocorticoids are released in a circadian and an ultraradian manner to control reproduction, skeletal growth, cardiovascular function, vascular tone, salt and water balance, a variety of things within the human body. These glucocorticoids are also especially important for maintaining homeostasis in the immune system. And this is why drugs that are synthetic analogs of these glucocorticoid hormones are termed these synthetic glucocorticoids. So these are the most commonly prescribed anti-inflammatory and immunosuppressive agents. But because the glucocorticoid receptor is distributed in several bodily tissues, these drugs have a wide range of side effects that affect many different organ systems. However, there is another use for these drugs that's not included on this visual because it just doesn't receive as much attention outside of a particular group of health providers. And so that group I'm referring to are OBGYNs. So in a healthy pregnancy, a developing human fetus is exposed to glucocorticoids that are produced either by the maternal or by the mother or by the fetus, and that depends on gestational age. So here we're looking at the first and the second trimesters. During this time period, the fetal cortisol levels are relatively low because one, the fetal HPA axis is underdeveloped, and two, the maternal contribution to the fetal cortisol content is low because of metabolism by this 11-beta hydroxysteroid dehydrogenase 2, which is a mouthful, so for short, 11-beta HSD2. So this is an enzyme with oxidase activity localized at the placenta that inactivates cortisol to cortisone. However, in the third trimester, placental expression of 11-beta HSD2 decreases, creating this surge in fetal cortisone content. In addition, increased secretion of CRH by the placenta creates a surge in fetal cortisol production by the fetal adrenal gland. So brain expression of 11 beta HSD2 also decreases with advancing gestation, and this is evidenced by mRNA expression in the mouse fetal brain. And so again, this endogenous surge of cortisol that occurs in the third trimester is necessary to promote rapid fetal organ maturation, as well as parturition, or the initiation of labor. Preterm birth complications are a leading cause of infant mortality. So in the case of either threatened preterm birth or preterm labor, which commonly can result from hemorrhages or severe preeclampsia, it's medically recommended that a single dose of these synthetic glucocorticoid drugs be administered to pregnant women to accelerate fetal organ maturation, although some clinicians opt to give multiple doses of this drug. So the two most commonly used drugs are going to be betamethasone and dexamethasone. And so these are 25 times more potent than cortisol. So they readily cross this placental barrier due to their resistance to the metabolism by this 11 beta HSD2 enzyme. And so these again help to mimic this endogenous surge of glucocorticoids that would normally happen during late gestation, but because these babies are going to be born early, we need to give them these excess glucocorticoids early. And this is especially important for lung development. So GR is expressed in the lung as well as in the brain at this time period. And we know that the most pressing issue for premature babies is just getting them to be able to breathe on their own properly. So this antenatal synthetic glucocorticoid therapy is standard clinical practice, and it is undoubtedly a life-saving measure associated with shorter ICU stays for these babies in the hospital. However, although they are clearly life-saving drugs, over the years, studies have made it more evident that elevated maternal cortisol content resulting either from stress or from exposure to these drugs disrupts glucocorticoid production during pregnancy, resulting in adverse neurodevelopmental outcomes. So clinical and non-clinical studies consistently report differences in glucocorticoid receptor dependent programming of three things, and that being the HPA axis, the stress response, and endogenous glucocorticoid signaling in the offspring. In humans, reverse adverse outcomes include, but are not limited to, attention deficits, executive dysfunction, and neurocognitive disorders in adolescence, as well as persistent HPA axis dysfunction in adulthood. However, other investigations found no neurodevelopmental consequence in these people, highlighting the need for standardized neuroanatomical and neurofunctional measurements to be made across clinical studies. So I wanted to highlight a particular large clinical study, and this measured the rate of suspected neurocognitive disorder in children aged 0 through 9 who were either exposed to the drug in the red column or not exposed to the drug in the green column prior to being born. So on the left, this is the study design here, showing the exclusion criteria, but what we really care about are these two groups at the bottom, either exposed to antenatal corticosteroids or not exposed. So the interesting thing is all of these babies were actually given these drugs for concern of prematurity, but then they were actually born to term, so they were carried for the full pregnancy. And this represents a population of children who received these extra glucocorticoids despite the fact that they ended up not needing them. So this bar graph shows the rate of suspected neurocognitive disorder in these children per every 1,000 people on the y-axis, stratified by age on the x-axis. And you can see that at many time points, children exposed to these drugs had a higher rate of suspected neurocognitive disorders. And this seems to be more prevalent in the younger ages. It may kind of level out or normalize itself over time. However, there is a debate over whether these children may be more vulnerable to stress-related diseases later on in life. So the longitudinal data on this is just simply still being collected in detail. So in the DeFranco lab, our mission is to identify the molecular signatures of glucocorticoid signaling pathways in the developing brain, specifically within the hypothalamus and the cortex, to understand how fetal exposure to synthetic glucocorticoids reprograms distinct neural circuits in the developing brain. Now, the biggest study which led up to my dissertation project was conducted by Dr. Crystal Fram. So she was a postdoc at the time in the lab who went on to have her own lab here at Pitt. And her mouse model focused on the effects of these drugs on neurostem and progenitor cells. So these cells are the multipotent self-replicating progenitors that make up the neural epithelium lining the lateral ventricle here. So during neural development, which is shown as a time course here, all neurons and glia arise directly or indirectly from these neural stem and progenitor cells to populate the cortex in a spatiotemporally coordinated fashion. RNA-seq gene expression analysis revealed that dexamethasone significantly and robustly changes the expressions of hundreds of genes in neural stem cells derived from both the fetal cortex and the fetal hypothalamus. So here we're looking at transcript expression of dexamethasone responsive genes with the log 2 fold change of transcript expression shown on the x-axis. The color indicates whether the gene expression changes occurred within the cortex or the hypothalamus. And these changes occurred in a highly sex-specific manner. So here we're looking at dex-dependent gene expression changes in males on the left, whereas in females are shown on the right. So this was the first RNA-seq characterization of glucocorticoid responsive genes in these specific cell types. But we know that if you look at other tissues or if you look at other cell types, some of these gene responses to glucocorticoids are shared in similar tissues. However, we know that if there are a subset of genes that are responsive to, say, dexamethasone only in the brain, or more specifically, only in neural stem and progenitor cells. So even here, you can see we have many more genes that are responsive to dexamethasone in the cortex as opposed to in the hypothalamus. And so what we wanted to know is, why do glucocorticoids regulate these specific genes, in particular in neural stem and progenitor cells? And to dive into that, we need to have a knowledge base of the key mechanisms of transcriptional regulation by the glucocorticoid receptor. So what is the glucocorticoid receptor, or GR for short, is a transcription factor. So when this receptor is bound by a ligand, such as cortisol or dexamethasone, or just glucocorticoids shown here, it will directly bind to sequence-specific elements of DNA, termed glucocorticoid responsive elements, to influence gene expression. In addition to direct DNA binding, which is shown here, in case B, it's well established that the receptor interacts with these cofactor proteins at nearly all gene regulatory sites. So this mechanism can allow the receptor to indirectly influence gene expression by using another protein to tether itself to the DNA. We also know that these cofactors can just simply exist nearby each other. And importantly, these cofactors are cell type specific. So this model helps to explain why the receptor binding patterns are cell type specific, as well as why we see cell type specific gene responses in different tissues. Now, all of these instances show the receptor binding to genomic regions that are located very close to their target genes. However, models of transcriptional regulation have helped us to understand that these transcription factors can also bind to distal elements located very far from the promoter regions of their target genes. And so when they bind to these distal sites, they can influence transcription through what we call this chromatin looping. So this occurs when these enhancer regions are brought into close physical proximity of gene promoter regions through what we call these looped chromatin conformations. So this mechanism can allow, say, GR binding at distal enhancer regions to regulate target genes that are located thousands of base pairs away, or even hundreds of thousands of base pairs away. As you can see in this multivalent model, chromatin looping doesn't always connect an enhancer to the nearest promoter. these loops can skip hundreds of protein coding regions to reach its target gene. And so when you look at this, you can see how models of transcriptional regulation get very complicated very quickly. Finally, models of transcriptional regulation tell us that chromatin accessibility restricts the receptor's access to DNA. So what is chromatin accessibility? So this refers to the degree of compaction of chromatin inside the nucleus. So eukaryotic chromatin is a complex of nucleic acids and proteins that exist inside the nucleus in kind of this really fluid, dynamic-like state. So about 145 base pairs of DNA are wrapped around an octamer core of histone proteins. And together, this makes up a single nucleosome, whereas when you string those nucleosomes together, that's collectively what we call the chromatin. Transcription factors and covalent modification of histones, mainly acetylation and methylation, are what I want to focus on today, dictate the conformational states of chromatin in this process called chromatin remodeling. So we know that tightly compacted heterochromatin is more likely to be transcriptionally inactive, whereas if you have this loosely wrapped chromatin, it's going to be more accessible to be acted upon by transcription factors and other machinery necessary for the initiation of transcription, meaning that this accessible chromatin is more likely to be involved in gene regulation. So based upon this knowledge, we wanted to map the regions of accessible and inaccessible chromatin in our neural stem and progenitor cells to determine which regions were more likely to play an active role in gene regulation. The second thing is we also wanted to determine if glucocorticoids change gene expression by remodeling chromatin at specific genomic sites. So to conclude this introduction, I've told you that GR is a transcription factor that binds to distal enhancer elements in addition to gene proximal elements, and that chromatin accessibility restricts the receptor's access to DNA. So the goal of my dissertation was to characterize the transcriptional regulatory mechanisms of GR in cortical-derived neural stem and progenitor cells. And so in the first aim, that's what we did. We characterized the chromatin landscape. So we use a mouse model of embryonic cortical-derived neural stem and progenitor cells, and these are grown as three-dimensional neural sphere cultures, shown here in panel E. We follow a precise microdissection protocol to isolate a specific section of the dorsal telencephalon, and these include cells in the ventricular zone as well as the subventricular zones. Now, with respect to human neural development, this mid-gestational time point, embryonic day 14.5, resembles the earliest ages that human fetuses are exposed to these synthetic glucocorticoid drugs, with respect to a time period of robust gliogenesis, ongoing neurogenesis, and then importantly, it's a low endogenous fetal and maternal glucocorticoid content. So this is about, in mice, this is about 2 days before the fetal adrenal gland starts to kick in, and about 3 days before the maternal content to corticosterone starts to rapidly increase. So once we establish these cells in culture, they're treated with 100 nanomolar saturating concentration of dexamethasone for a total of 4 hours, or a vehicle control for the same time period, after which they are immediately collected for analysis. So we performed an assay called ATAC-seq. So this stands for Assay for Transposase Accessible Chromatin. And this is a technique that utilizes a bacterial enzyme, this TN5 transposase, is an enzyme that is simply able to access more loosely wrapped DNA, whereas if it's inaccessible chromatin, this bacterial enzyme just simply cannot physically access those regions of the DNA. So once it finds those regions of accessible DNA, it can tag those fragments, and then we can purify those fragments, amplify them up in a PCR reaction, and then sequence them, resulting in what we call these ATAC-seq peaks, where if you have a bigger ATAC-seq peak, it means you had more sequence reads for that specific spot of the genome, meaning that that region was more accessible when compared to others. And so this schematic on the right here is what we're going to get as our output, where all of the ATAC-seq peaks that we used in our analyses met the threshold for significance, which is a p-value less than 0.05. And again, this will allow us to see if we have accessible chromatin that's not affected by dexamethasone, or if it is remodeled by dexamethasone at specific sites. So this analysis revealed roughly 40,000 regions across the entire genome, the majority of which were located within intronic or intergenic regions. So these represent either enhancer or promoter regions that are physically permissive to interactions with transcription factors. Now, despite this rather wide distribution of accessible chromatin across the entire genome, if you focus only on the protein coding region, so only on genes, a heat map of sequence reads shows that accessible chromatin was densely distributed near the transcriptional start site of most genes. So these accessible regions of chromatin, in addition to being accessible to or open for transcription factors to act upon them, they're also located where you typically have the machinery necessary for the initiation of transcription. So, for example, RNA polymerase II. So this is data showing accessible regions of chromatin in each of our triplicate treated vehicle or dexamethasone treated samples. And our replicate values were extremely tight. So we had a really beautiful data set to work with here in terms of limited technical variability. So this is located at a specific spot in the genome. This is the transcriptional start site indicated by the green line of HIF3 alpha, which is a highly dex responsive gene in several different tissues. And again, the biggest question we wanted to answer was, does most of our chromatin look like that indicated by the red bracket where it's not changed by dexamethasone, or does it look like this instance here on the left indicated by the blue bracket? A Pearson's correlation analysis reveals that total accessible chromatin was highly correlated between treatment groups. So our Pearson's mean R value was 0.97, whereas a value of 1 indicates perfect correlation between treatment groups. Of the roughly 40,000 accessible regions, only 95 of them were differentially expressed between treatment groups. And this is represented by the 95 points on this graph here, where the x-axis is showing the fold change of ATAC-seq peak read intensity, and the y-axis is showing the p-value. So all of these points were statistically significant. And so we wanted to ask again, where do most of these regions of differentially expressed chromatin occur in the genome? And most of them, again, occur at intergenic or intronic non-coding regions. And these are far removed from promoters. So this prevents us from examining how chromatin remodeling directly impacts transcription or cell fate outcomes. So with respect to chromatin biology in our neural stem and progenitor cells, we learned 3 key points from AIM-1. The first is that majority of accessible chromatin occurs in distal non-coding regions. And again, it's hard to predict the functional relevance of these sites. However, when we combine our data from AIM-1 with the data I'm going to show you in AIM-2, we'll start to gain a better understanding of how these non-coding genomic sequences can shape early neural development in some of these kids that are exposed to these excess glucocorticoids before being born. Secondly, we learned that most of the chromatin landscape is not altered by dexamethasone, indicating that this chromatin is accessible, it's ready for the receptor to bind to it, even before you introduce this drug into the culture system. And thirdly, we learned that DEX triggers chromatin remodeling at 95 unique sites. Future endeavors which profile 3D chromatin looping might help to provide some valuable insight as to why this remodeling occurs specifically at these 95 sites to facilitate a gene response. So in AIM-2 of the project, we went on to define the genome-wide GR binding patterns to learn more about how dexamethasone drives transcription. So we use the same experimental setup and we performed ChIP-seq, which is a chromatin immunoprecipitation assay. And so this assay will allow us to determine if we have in case A here, glucocorticoid receptor binding in chromatin that is always open, if we have glucocorticoid receptor binding in the presence of a ligand that maybe induces some chromatin remodeling, shown here in case B. We can also look at where you have regions of accessible chromatin in case C, and the GR does not find this region at all at this time point, as well as if you have accessible chromatin all of the time in case D, but you need the addition of the ligand for the receptor to interact with that specific spot of the DNA. And so this ChIP-seq technology relies on the fact that you have DNA that interacts with proteins. And so you can cross-link these two things together. And then after you cross-link these two things together, you can use a reliable antibody against the glucocorticoid receptor to isolate the DNA fragments that are associated with the glucocorticoid receptor, after which you can purify those fragments, amplify them up, and again, sequence them to look at where the glucocorticoid receptor binds to specific genomic regions. Now, the most important thing to note about this technique is that in addition to isolating these DNA fragments associated with GR, we can also identify transcriptional cofactors or co-regulators that are closely associated with GR in the same nucleated complex. And so this will help us to identify maybe some of those transcriptional co-regulators that guide the genomic response to dexamethasone, specifically in our cell type. So this is our real data overlapping ATAC-seq regions of accessible chromatin and GR binding sites here on the bottom at a specific region in the genome. So in total, we had over 3,000 regions of over 3,000 GR binding sites in our dexamethasone treated cells. And the genomic distribution of this looked really similar to that of our accessible chromatin, where most GR binding occurred in intergenic or intronic non-coding regions. When comparing vehicle and dexamethasone treated samples, we had 941 regions bound by GR in vehicle treated samples, where the addition of this synthetic glucocorticoid brought that number up to over 3,000. Now, if you look at subgroups within the vehicle or the dexamethasone treated samples, you can see that GR binding occurs mostly within this open chromatin as opposed to within the closed chromatin. Our results agree with findings in other cell types which show open chromatin in distal genomic regions as a biological prerequisite for most of glucocorticoid receptor binding. So next we went on and we performed multi-omics data integration by combining our ATAC-seq data and our ChIP-seq data with some publicly available data sets. But this yielded quite a bit of interesting and reportable data, but I want to focus on what we learned that is the most biologically relevant. So we were interested in the number of GR binding sites that occurred at distal enhancer regions. And we know that these distal enhancer regions are marked with an enrichment of this H3K27 acetyl histone modification marker. And so these regions with an enrichment of this histone modification marker would be candidates that were involved in chromatin looping and gene regulation. So we used histone modification marker data, and these were collected in the exact same cell type at the exact same embryonic time period to determine if glucocorticoid receptor binding occurs in some of these distal non-coding regions with an enrichment of this histone modification marker. And again, these would be candidates for being involved in chromatin looping. So to simplify that, these are the three data sets that we combined. In both treatment groups, we saw that GR binding quantified by the number of ChIP-seq peaks on the y-axis preferentially occurred at accessible chromatin that had an enrichment of this histone modification marker when compared to accessible chromatin either lacking this histone modification marker or simply GR binding at regions that were inaccessible. So these data corroborate the notion that the glucocorticoid transcriptome is controlled not only by promoters and cis regulatory elements, but also through GR responsive distal regulatory elements. Now, we still don't know which genes that these distal enhancers regulate, but the collection of chromatin looping data is the logical continuation of this project. So this would allow us to investigate if drug exposure influences cell outcomes and eventual neurodevelopmental outcomes by modifying some of these enhancer promoter interactions that regulate the expression of neural determinate genes. Next, we utilize motif enrichment software to predict the transcriptional co-regulators associated with GR in nuclear space. So this is a standard part of processing for ChIP-seq data that applies an algorithm that searches for specific nucleotide sequences under our ChIP-seq peaks. So this program has a library of specific nucleotide sequences that are known to bind specific transcription factors. This analysis revealed an extremely high enrichment of our glucocorticoid responsive elements under our ChIP-seq peaks. That's the first thing we should see. If we see anything besides a GRE, it might raise some questions. And the motifs of these top 3 rows are near identical. So all top 3 rows here indicate instances of GR binding directly to glucocorticoid responsive elements. The other significantly enriched motif that we found of interest belonged to the SOX family of transcription factors. And this is where the project takes a turn. This was quite a surprise because studies in other cell types do not identify SOX transcription factors as a cofactor for GR. So this suggests that this aspect of glucocorticoid action is specific to our cell type. So the results of the motif enrichment analysis are quantified here. So in vehicle-treated samples at GR binding sites, about 33 of those peaks had at least one or more SOX motif, whereas less had a GRE motif, and the addition of dexamethasone increased those percentages in both groups. Now, we noticed instances where you had at a single GR binding site, you could have more than one GRE or more than one SOX, or a combination of the two. And so we wanted to know how close together were those motifs located? Were they very, very close together or were they far apart? And so we determined that when you had a GRE and a SOX motif at the exact same GR binding site, they were more likely to be located less than 100 nucleotide base pairs away from one another. And the number of occurrences where this happened outnumbered the total number of GR binding sites, again, because you could have multiple of these motifs under a single peak. These data suggest that in addition to direct DNA interactions, GR activity preferentially occurs at sites that are co-occupied or at least closely associated with the binding of SOX transcription factors. So from AIM-2, we learned that SOX transcription factors are a putative cell type specific transcriptional cofactor for GR in our neural stem and progenitor cells. Now we know that these SOX transcription factors are of relevance to gene regulation in our cell type. So landmark studies by our collaborators in Italy have shown that SOX2 maintains over half of all enhancer promoter interactions. So it's very important for maintaining chromatin looping and gene regulation, as well as we know that SOX2 shares over 145 target genes with GR. And so we wanted to look into this further. The first thing we did in AIM-3 was we conducted a biochemical assay. This is a proximity ligation assay to measure the protein-protein proximity of GR and SOX2 in culture. So how this works, this is a biochemical assay that utilizes two primary antibodies raised in separate species against two proteins of interest. So this would be GR and SOX2. The secondary antibodies have PLA probes, which when they're located very close together in culture, less than 40 nanometers apart from one another, they form this DNA circle that undergoes this rolling circle amplification. And this can be detected by fluorescently labeled complementary oligonucleotide probes. So that fluorescent signal can then be visualized by confocal microscopy. So here, each of these red dots in the first two panels indicates instances where you have these two proteins located close enough together to suggest that they exist within the same nucleated complex. When you quantify the intensity of these probes, you see that the average probe intensity was similar between the two treatment groups, whereas the negative control groups had significantly reduced amounts of probe photoactivation. However, you'll notice if you look at these two panels on the right, by eye, there was no photoactivation. We then asked what percentage of cells have these potential protein-protein interactions. And so when we quantified that, about 62% and 40% of vehicle or dexamethasone treated cells had this potential protein-protein interaction. And so together, these biochemical data support our bioinformatic prediction of these two proteins interacting within the nucleus to coordinate gene responses. Now, to test this further, in our final set of experiments, we utilized the transgenic SOX2 knockout mouse model to investigate SOX2 as a transcriptional co-regulator of GR. So this is a technique that allows you to delete SOX2 using a Cre recombinase in Nestin-positive neural stem cells. So we established these cultures at postnatal day 0. And at this time, neurogenesis is not significantly altered in vivo, whereas in culture, these cells retain the ability to self-proliferate up for about 10 passages, or about 30 days in culture. The proliferation rates are comparable between these two mouse strains, but after which in the SOX2 knockout cells, you will start to see a decline in the proliferative capacity. So we treated these cells with the saturating concentration, 100 nanomolar concentration of vehicle or dexamethasone, again for 4 hours. And we did this after passage number 3, when the cells are still proliferating nicely, after which we performed unbiased genome-wide measurements of gene expression by microarray. This revealed that dexamethasone significantly alters the expression of 429 genes in our wild type cells, whereas in our SOX2 knockout cells, that number increased to 901. Now, the fact that the number of dexamethasone responsive genes dramatically increases when you remove SOX2 from this culture system suggests that there is a potential de-repression of glucocorticoid receptor action. This allows us to build a hypothetical framework of transcriptional regulation that includes chromatin looping. So moving forward, we can use ChIP-seq technology along with this chromatin conformation technology to test whether SOX2-mediated chromatin looping connects some of these distal GR binding sites to genes that are responsible for neuronal function. So when we looked at the highly DEX upregulated and the highly DEX downregulated genes, some genes were that response to dexamethasone was shared between the wild type and the knockout groups. And this suggests that that gene response occurred independently of SOX2, because it didn't matter if SOX2 was there, it didn't matter if SOX2 was ablated. In contrast, another subset of gene regulation occurred uniquely in the wild type group. This differential gene response suggests that a subset of GR target genes are SOX2 dependent, because when you remove SOX2 from that culture system, the gene response does not occur or is changed. Lastly, based on gene expression signatures in our data, canonical signaling pathways may be differentially activated by dexamethasone, dependent upon SOX2 function. So here we're looking at biological signaling pathways that are either activated, indicated by orange coloring, or repressed by dexamethasone in our wild type or our knockout cells. So not all of these pathways are specific to brain function, but a few do stick out as being essential mediators of healthy brain development. So these data tell us that SOX2 may be critically involved in restricting activation of this endocannabinoid neuronal synapse pathway, as well as this dendritic cell maturation pathway. SOX2 also might be involved in preventing this repression of the growth factor FGF signaling pathway in these neural stem and progenitor cells. The role of SOX2 in directing glucocorticoid action in humans is still very understudied. However, our data suggests that these glucocorticoids, or that SOX2 influences glucocorticoid action during these really early neural developmental time periods. But it's also unknown whether glucocorticoids impair SOX2's ability to maintain chromatin looping in the downstream gene responses. So our genomic data may advise clinical follow-up studies in persons exposed to these synthetic glucocorticoids in utero. So we don't know if long-term or epigenetic programming occurs that makes these persons vulnerable to psychiatric diseases that have a SOX2 related etiology, some of which are listed here. I also mentioned in the beginning of the talk that synthetic glucocorticoid therapy is often used to address inflammation and the inflammation and immunosuppressive and the immune system. And these drugs are at times recommended for people that have a CNS disease that are caused by genetic SOX2 polymorphisms or mutations. So our data may inform the health risks that are associated with giving drugs to these populations of people, because although they're trying to minimize inflammation, we don't know if they could be exacerbating disease onset or disease symptomatology. We also know that either exposure to these synthetic glucocorticoid drugs or early life stress cause similar health outcomes in terms of persistent HPA axis dysfunction. So future studies of stress-related mental disorders should consider this interplay between the glucocorticoid receptor and SOX transcription factors as they develop new theoretical models of disease pathology. So in summary, we found that excess synthetic glucocorticoids do have the potential to alter GR transcriptional responses by acting upon a developmentally regulated chromatin landscape to influence the gene expression of our neural stem and progenitor cell populations. And ultimately, this might contribute to a cascade of biological changes that lead to some of these adverse neurologic outcomes. Lastly, the continuation of this work may ultimately inform safe clinical applications of these corticosteroid drugs. So I have a few acknowledgements, obviously, Don, from the start. I really appreciate all your mentorship. It's been an amazing ride, and I am sad to see it end, believe it or not. And everyone in the DeFranco lab, Crystal, Fram, Lexi, Franks, Julianne, Kai, Li Ping, Laura, you guys have been so helpful in all of my experiments and listening to my frustrations and everything. Natalie, our newest addition, I love having all of you guys in the lab. You've been so helpful. I'd like to thank all of our collaborators at Pitt, including Dr. Uma Chandran and Feng Ping. They did a lot. They were a part of our bioinformatics team. So a lot of brainstorming, a lot of time spent with them. as well as our external collaborators. Paula Monahan Nichols is our collaborator for how many years? Many years, Don says, a long time. As well as Inez Ragazzi at Cornell and Silvio Nicholas in Italy. You guys have been instrumental to getting all of this data, all of these experiments to work. So I really appreciate your guys' mentorship in this process, as well as a big thank you to my thesis committee and your guidance and the oversight that you guys have provided. I'm really looking forward to our discussion after this talk. And then finally, friends and family. I'm not going to name them all, but you guys all know who you are. You guys have literally held my hand through this whole process. So I really appreciate you guys. And again, this thesis is dedicated to my dad, who is the head of our family and he's always stressed academic excellence and really excellence in everything that we do and doing everything with integrity. So this is dedicated to you, Dad. And thank you, Mom. Yes, any questions? I guess.
Thank you so much, Kim. I just want to remind the committee that all the questions, but people in the audience have questions.
So when you, at the beginning, you talked about the difference between males and females, when you went through the ChIP-seq data and all that data, was that males and females?
No, so all of our genomics data so the RNA-seq data was males and females. All of our genomics data after that, so the ATAC-seq and the ChIP-seq was all in females. So we started with just the one. With Dan said money, yes. Money and resources are a big one, but that's definitely like a future direction, is to include the comparison between males and females to see, do males and females have differentially regulated chromatin landscapes? Is that why we see differences in the gene expression patterns? And interestingly enough, in clinical data, they also see differences between the males and the females and the neurodevelopmental outcomes as well. So that's a big piece of it, that we're only looking at one side of it to start off. Yeah. Brian, go ahead.
In the Poker study, are the decks there all the time?
No, so we only added DEX for 4 hours. So yes.
Can you do any time point data that tells you how persistent the GR effect was going out in different times, or something like some genes affected for a long time and others very cheaply?
Yes, so no, we did not do that, but we do know that there are primary gene responses that occur, say 6 hours and prior, and then secondary gene responses that occur after 12 hours that require new proteins to be translated and all of that. So there is we're only looking at one time point. And so doing this at multiple time points, which would require 4 more of me, would provide more information into the regulation. Yeah. Yeah. Yeah.
So when last aim, when you're looking at the potential functional relevance of the SOX2 index, you have the suspected downstream effects of the gene regulation. Do you guys look at if you just let the knockout models or biotechs develop with the DEX exposed, if there would be a configurable effect to it or some sort of observable?
Yeah. So the question is, if you if you let the SOX2 cells just grow. Okay, if you treat a SOX2 knockdown animals and just with dexamethasone and just let them go, do you see behavioral deficits or something like that? So a SOX2 knockdown is lethal after a certain point. So the animals that don't last very long after the first week of life. So that's one of the caveats. So it's helpful to look at early neurodevelopmental time period, so before birth. But after birth, it is, if you have the full knockout, it's lethal. So that's kind of the answer. Sorry, the other questions for the people in Zoom before that, Julianne asked about males and females. So the RNA-seq gene expression data was collected in males and females, but a lot of our chromatin accessibility and our GR binding patterns, we only looked at females to start off, but we do suspect that we would see differences between males and females based on the differences in gene expression, as well as we see in clinical populations. Males and females have differential neurodevelopmental outcomes in humans. Paula, do you have a question?
Yes, I do. Great presentation. First, I want to say great presentation, Kim. Extremely clear. Wonderful data. I had a couple of questions. The first thing was, what is known about the other proteins that SOX2 interacts with the neural stem cells? And can you envision a role for them in, DEX regulation of neural development based upon what you know about the biology of the cells?
Yes, that's a great question. So there are a lot of other similar bioinformatics studies that kind of follow the same pipelines that show that SOX2 has its own cofactors, and these cofactors change over time. So SOX2 can interact with cofactor A in neural stem progenitor cells, but then it'll switch out cofactor A for cofactor B when it controls gene expression, say, in newborn neurons. And so there haven't been a lot of studies that see if SOX2 and GR share cofactors, but that would simply be another next step, an important question to ask. And you could even start just by looking within the literature. But because this is the first study of GR in neural stem cells, and we only saw SOX transcription factors, we didn't see any others that SOX shares the same cofactor with. So it would be interesting to do like co-immunoprecipitation studies or something like that with candidates that SOX2 and GR might share, but I'm not saying I have to go, so.
On a related, just to follow up to that, in your ligation assays,
Mm-hmm.
the cultures, when you plate them, they have mixed cell types, mixed progenitor types, and I wondered if, in looking at these cultures, whether you saw a predominance of signal in a particular morphological type of cell. It looked like the ones you showed, they were all the big neural stem cells. So I just wondered whether you always thought it was the big ones. Some of the small ones are usually oligoprogenitors.
Mm-hmm.
I was just curious, you may not have looked at that at all, but I was just curious if you saw a predisposition of signal in a type of morphological progenitor.
Yeah, it's not something that we looked at specifically, so we didn't add an extra stain to look at morphology. When I was looking at the images, I myself didn't notice anything that was, consistent across slides. But it would be a second question that we have the same images. We could go back and look at morphology as well and try and look at that in a different way. But no, I didn't notice anything drastic by eye. Yeah. Yeah. Thank you. Thank you.
So we moved you on
Mm-hmm.
work in the culture group, we see that there was some staining of fluorescence outside of the gapping.
Yes. Yes. So the question is, in the proximity ligation assay, we noticed that it seemed to be that there was some staining of those puncta outside of the nucleus. And what is that? We've had that conversation several times. We believe, yeah, yes. Yes. Yeah, so in the literature, they do say that SOX can be cytoplasmic. In my own personal staining, SOX is always nuclear. So we believe it's just a function, like an artifact of the assay. It involves a lot of washing and things like that. That's my answer. Yeah, it's a good point. We've debated it, but all the quantifications are just of those puncta within the nuclei. So those in the cytoplasm were not included in quantification. Mm-hmm. Yes, yeah. So when we have 5 more chems, then we'll dive into that deeper. Yeah. Yeah. Another few years.
You don't have an engineering question? No, good. Thank you, everybody, for coming. Tim, congratulations, great talk, and yeah. You're all invited.
And the conference room, we have it at one.
One? Okay.
But I'll see if I can, we can get
Yes.
Okay, thank you guys all for tuning in.
Well, I guess John on the show now, right?
Yes, Dr. Sorrells, Don just said, all right, Sean's running the show. It's his turn now.
All right.
Thanks,
Sounds
Paul.
good.
Yeah, can we explain?
Yes, yeah, we're gonna,
I think my role
yes.
Yeah, so Dawn is supposed to not say that much.
Okay, so that concludes the seminar. So everyone else that's not on the committee, if you could kindly log off, I will all talk to you later. Love you, family. Okay, yeah. All right. Oh,
Okay.
yeah. Yes.
All right. So I think that the next point here is for us to spend some time discussing with Kimberly the exciting findings that she's made. I'm sorry I can't be there today in person. And thank you, Kim, for helping us to see you and your data in the room as best as possible. So we basically have time now to just kind of go around and ask questions, and I can start us off maybe by asking a couple of questions that I got in my thesis defense when I presented work on glucocorticoids, and they still haunt me to this day. So you get to carry them forward.
Give me 1 second. I'm trying to put you
Sure.
on our projected screen so I can see you.
Oh, okay.
Yeah, give me 1 second.
you can't get it.
You can just bring him in, but if you want to, you can mill that thing over, that will reach,
Will it reach?
or we could grab,
Well, you don't need to be plugged in.
we could go around. We can come over there and sit around it.
Yeah, I'm not exactly sure. So
We can see it.
there's a little
Yeah,
table
it's-
set up.
I think it's fine.
We're good.
Trying to.
okay. I can see everyone. Hi, everyone.
Well, at me, but I'm not here.
Okay.
Oh yeah, Don.
But I'm not
Okay.
here.
All right, that works pretty well.
Okay.
Okay, so my first question for you is, I mean, it's really exciting seeing it all presented all together like this. And one of the first things I kind of keep coming back and thinking about with glucocorticoids, especially is concentration. So I know that, you know, the 100 nanomolar concentration that you selected, there's only so many different doses that you can look at. So I wondered if you could speak a little bit more to the rationale for that dose and what you think might be what you would see essentially if you looked at different doses, because glucocorticoids, as you know, are really exquisitely detected in different concentrations by the whole body, by the receptor systems that detect them. So basically the first question is, you know, why did you pick that dose? And the second question is, how do you think it would be different if it was a different dose, higher or lower?
Yeah, sure. I'm moving a little closer so you can hear me. Okay, so the 100 nanomolar concentration of DEX that we use, as I mentioned in the talk, it's a saturating concentration. And we chose that concentration because we wanted to compare our gene expression data with the chromatin accessibility data, et cetera, at the exact same concentration. So that's why my experiments use that concentration. Now, we know that in culture, that's what we consider more of a high dose of dexamethasone. So we've also done, previous studies have done a lower dose of dexamethasone, and we see that there's a dose dependent response in proliferation for those cells. So at the high dose concentration that's used in these experiments, we see a decrease in proliferation in culture. But if you have the low dose, we see an increase in proliferation in culture. And so comparing that to gene expression, we would definitely see differences in the low dose versus the high dose in genes that are controlling cell cycle, apoptosis, et cetera. And if you want to compare that to humans, there's not a direct comparison of the dose, the concentration of dex in the fetus and how you can approximate that in culture. But we know that in humans and in mice, if we give the dose in vivo, you see an increase in proliferation. So that maybe more mimics the low dose that we would use in culture as opposed to the high dose that's used in these experiments. But yeah, there's not an exact, calculation for if it's this concentration in humans, we use this in cells, but we use a saturating concentration to look at the primary gene responses. Yes, that's my answer.
And then the second part of that is about timing. So I know that you selected one time in embryonic development to look at these effects, and then essentially one time in postnatal life as well. But do you think that there would be like, how do you predict the differences? How would you predict different outcomes based on looking either with an earlier treatment of glucocorticoids, or because I mean, some of the thing too is again about trying to build this clinical relevance. The time window that embryos are usually experiencing this in the human is a little bit later maybe than what you're selecting in mice. At least from, I mean, it can be hard, right, to make a cross species.
The range in humans is wide, so it's anything up to like 30 gestational weeks. So it could be, at 22 weeks, it could be at 28 weeks. It varies.
Yeah. So do you think it would be a more profound effect earlier or later? And I guess.
Yeah. So the major point about that question in terms of timing is what are the levels of fetal endogenous corticosterone levels in mice or cortisol levels in humans? That's the main thing. Because if it naturally increases later on. And so we know from clinical studies that if you give the dose of the drug and more than 7 days go by before that baby's born, they're going to have worse consequences than if you give them the drug and they're born immediately. So it also depends on how long it's present in the system and when they're born. But in animals, I would expect that at earlier time points, the deficits are going to be more drastic when compared to later time points, just because of the timing factor. And it's going to naturally increase, towards the end of gestation. So the impact is going to be less adverse than at earlier time points.
And what about the different cell populations that are dividing at different times? The early expansion of the progenitor pool versus later neurogenic divisions. Do you think that those would be differentially affected based on your data?
Yes, absolutely. I didn't show it in here. I could pull up a slide, but I'll just tell you about it. So when we give the dose at this embryonic day 14.5, there are previous studies showing that it has a maximal effect on the proliferation of cells that are the neurons migrating to layer 5 of the cortex. And we know that, neurodevelopment is very temporally controlled. So if you give that dose at a later time, it's probably going to be more upper level neurons. Whereas you give it at an earlier time, I would definitely predict it would be primarily, say, layer 2, 3, as opposed to layer 5, just because what we know of the timing of where these neuroscience progenitor cells are destined to be, day by day by day by day. Does that make sense?
Sorry, what was, so the earlier ones you think would be which cortical layers?
More deep cortical layers, and then later on more superficial.
Okay, got it. Yeah, that's great. So I have many more questions, but I'm gonna let us, maybe we can each go around and ask a couple questions, starting off with whoever feels like they have a burning question in their pocket.
Maybe I can go second.
Sure.
Great presentation, Kim. So I just have few questions about your analysis because I guess I covered that territory. So you said you identified 95 differential ataxia peaks, and most of those peaks, and so all of those peaks were intergenic and intronic regions?
Not all of them, most of them, the majority of them. So there were a few, say less than maybe 8 or 9 that were near a transcriptional start site, but yeah, most of them were distal. Yes.
Okay, so if for example, you showed an example where you had the peak that became more accessible in the intron of HIF1 alpha,
Yeah.
and how does it affect the expression of HIF1 alpha?
So HIF1 alpha in neuroscience progenitor cells is highly induced. So I do, can I pull up figures? Can I pull up visuals?
Yeah, absolutely.
Okay, perfect. I guess I'll just pull up my dissertation. So, figures. Okay, so this is the example that you're referring to.
Mm-hmm.
Right. And so we did publish, so this is in my published paper, as well as this is in my thesis. So these were the differential peaks located, what you're referring to, added protein coding region or nearby the transcriptional start site. And so we did publish, so HIF3 alpha, you can see, I'm sorry, my head is blocking, is highly induced. And that's specific to that gene. So most of the genes were upregulated in those specific cases that you're talking about. There's one here that was downregulated. But it's interesting because it was downregulated, but the chromatin was made less accessible as opposed to more accessible. So maybe there's like a directional pattern there. But yeah, so we did look at those individual cases and we published them. Yeah, so most of the genes were upregulated.
Okay, so and when you showed us the ChIP-seq data where you did the motif analysis and you showed the motifs for the glucocorticoid receptor and SOX2, it appeared that the core motif is the same for both of them. So seeing SOX2 wasn't that surprising to me because the motif, the sequence looks very similar in both of those cases.
For a GRE and a SOX?
Yes.
Yeah, I mean that could explain why they're binding to the same regions as well.
Exactly. So the fact that people have not looked at SOX, haven't seen SOX2 at those loci would be a little surprising because the core motif is pretty much the same.
Yes, and that could explain why maybe you have like that tethering mechanism where there's a SOX motif where there's a GR and then a SOX and then the DA underneath because they're just attracted to the same nucleotide sequences. So that makes sense. Yes. So Stu saying that if you, no, you're totally fine. I'm sorry. I'm trying to go back and forth here. The GRE motif and the SOX motif, if you look at the nucleotide sequences base pair by base pair, they look very similar in terms of the representation of the core, the core letters that make up where that receptor is attracted to. So the motifs look similar is what she's saying, the representation of the base pairs. Yeah.
Just one more question from
Yes.
me. So if you look at your differential attack-seek peaks, are they enriched for any motif binding sites? Any known motif or any transcription factor?
So we did not do that analysis. We only did it for the ChIP-seq peaks. So we didn't look at it. We did not look at that.
That would be something like doing a differential version of that, seeing if there is something enriched in those although 95 is actually not a big number, so you might not pick up.
That's the thing I was going to say is I'm not sure, maybe we did talk about with our bioinformatics team, but to look for an enrichment, 95 might not be enough to look for that.
That's correct, but maybe just like a differential version of it where you do not see any differences, the peaks where you don't have significant variability versus the 95 might give you some indication of a transcription factor. Maybe it's not significant.
Right. Yeah, we did not. Oh, I'm sorry. We did look at that. Let me pull it up. We did look at it. Let me backtrack. So this is all sites. So we did look at it. I apologize. So, okay. Can you see this slide?
I can.
Okay, I'm sorry. So this is our differential attack-seek peaks, only the 95 sites, and we ran that motif enrichment analysis. So we did see GREs. This is all differential attack-seek peaks in the black column. And then we broke it down to say the increased accessibility or decreased accessibility for the chromatin. And so we did see that most of the peaks with increased accessibility had a GRE, a SOX, or both, whereas the peaks with decreased accessibility only had a SOX motif present. And then we tried to kind of come up with some type of visual to show that when you have dexamethasone that makes these peaks bigger, you have both motifs present there, whereas if you have dex that kind of closes the chromatin, there was only
So I think what I'm trying to get at, what did you use as a background when you did this kind of differential, the motif analysis? I wanted to see the peaks that are not different versus the peak that are different. What is the TF that is going in the peaks which are different? So an analysis where you don't choose the regular random background, but the peaks which are not getting differentially affected. Do you understand?
So I believe I'm understanding your question. So we only compared our differential peaks to a set of random ChIP-seq data that the program does as default.
Yeah, so that's why if you try a different background where your background will be the peaks that are not affected in terms of their accessibility, if the glucocorticoid shows on its own, that would be really confirming that this accessibility is really being affected due to the dexamethasone treatment.
Yeah, so we didn't do that final confirmation, so that's not something that we looked into, but I understand what you're saying.
Yeah, all right, that's it from me right now.
Thank you.
So I guess I'll start with sort of a technical question, clarification maybe. And can you guys on Zoom hear us in the room?
Yeah.
So for AIM-1, it's my understanding, and so that's what I want to just see if I'm understanding correctly or not, that you ran a vehicle and a DEX group in technical triplicate, but did you have any biological replicates?
No.
So this is one animal?
Yes, this is one animal technical triplicates. Yes.
Okay, and so are you concerned that things could just be very specific based on a single animal? And again, we're getting back to the question from the audience about it was just a female, so obviously male-female differences, but any other random effects that could have just been like the fetus you happen to choose?
That's possible.
Is that true? I'm sorry. Is that true? I thought we had different animals.
It's different animals for the ChIP-seq. The attack-seq is the same. Yes.
It's a little concerning to have an N of 1 per group. Like not having any biological replicates. So it's just like a caveat to, you never know if there's something could have been like weird about that. But So, and then I sort of getting back into the, a little bit what Sean was talking about of the, clinical relevance of translation. I just like to give it like a better understanding of what's known. So you were highlighting the one clinical data set where all the babies were born to term,
Yeah.
but some had that early
Yes.
exposure to DEX. So what happens to them after is it known if the cortisol response in late gestation is the same or different?
Oh, in late gestation?
Yeah.
So to my knowledge, giving these, that's a good question. I don't know. I don't know of studies that have looked at if you give a mom dexamethasone and then look at her contribution to the fetus at right before they're born, say gestational week 30, if that's different. I have not seen a study that measures that.
So, I mean, it's essentially, well, and this gets, broader about the literature on this, because I'm not aware of it, because that seemed to be like, it gets a lot harder when you actually look at the ones who were born preterm, because then you have the confound of like just being a preemie baby versus.
Yes,
versus
yeah,
not.
and that's a big problem with a lot of the clinical studies, is there are a lot of, I don't want to say confounders, but there are a lot of factors. So these are all simply associations. So some studies try to control for if babies are born early and they have like all these extra medical procedures and shots that, raise their stress level. So some studies do try to control for that and still show a direct effect of these drugs on, say, their cortisol response at age 1 or age 2. But you're highlighting a large problem or a large consideration when trying to like compare this study to this study to this study. They're just not standardized.
Right. I mean, I guess what I'm trying to get at is, are you modeling like DEX exposure, in a, developing fetus that is born prematurely? Or are you modeling the one, like the case where that might have gone to term and been born anyway?
So we're
born.
only modeling in this model the acute gene expression changes that we don't know how, if they were born early or born to term, would affect the outcome. So we're only modeling this acute response. So it's not really incorporated into like our experimental model to look at the difference between those two.
And then I was curious, so getting at the cortisol response again, is if you would see, or like I guess, has anybody like modeled like later gestation, like cortisol effects on like some of the genomic things that you're looking at? Like, so is there a difference between like just be based, would cortisol doing what it's naturally supposed to do have like a similar effect on like the accessibility of chromatin, et cetera, that you're observing?
So you're saying,
like, is it actually like the normal response, right? Like that you should get if, in late gestation, you're exposed to cortisol from the mother.
Is that response? So how similar is the response of dexamethasone to cortisol?
Yeah.
So dexamethasone binds GR much tighter than cortisol, but again, we can't have a direct comparison between the contributions of dex to the contributions of cortisol during late gestation. But the dexamethasone binds GR much tighter. So if you were to compare the same concentration of cortisol to the same concentration of dex in culture, dex is going to be elicit a much stronger gene response. So no, it doesn't exactly mimic, dex doesn't exactly mimic the cortisol surge that happens during late gestation, but that's not what we're trying to mimic. We're only trying to mimic these drugs at this mid-gestational time point. So it's a little bit, it's not what our data shows. Yes.
It's kind of look to see if that kind of thing. But I think that's all.
Well, I was gonna ask the same. And the same as Kathy asked about the cytoplasmic.
Yes.
Okay, so let me make sure I'm getting this correct. So HIF3 alpha is increased in response to that.
Yes.
So HIF3 alpha is one of those oxygen deprivation induced genes, yes? HIF1 alpha usually is this the same? Okay, so it's not,
It's,
Doing the
it's not a
same thing?
it's not a hyperoxia.
okay, that was what I was gonna ask, because I was wondering if this is mimicking hypoxia by giving corticosteroids. No?
I can't answer, but
Okay, that was, one question. The other is more of a comment that with increasing use of fetal MRIs, physicians are beginning to identify structural damage in fetuses that have a higher probability of having preterm delivery later.
Yes.
So that the trigger for preterm birth may originate in the fetal nervous system rather than in the mother.
Okay.
That's, I don't know how much evidence there is for that, but because the use of fetal MRI is increasing, that's becoming like a predictor of the risk for preterm birth. So
it's cool.
it's cool, but it also allows you. There are different examples of structural things that you can see around on MRI are evident in fetuses before they give birth to preterm.
Do they do this MRA like through the placenta, like while they're still in?
Yeah,
Oh,
yeah,
okay.
yeah,
Wow.
but then they go, there is a way of detecting the structural differences that is predictive of subsequent preterm labor.
It could be indirect though, because there's a lot of metabolomic studies now looking at predictors, biomarkers for preterm.
Yeah,
And there's microRNA and exosomes that are different, and there's other.
right, but there's also a contribution to the fetal brain.
Yeah.
Okay, so there are lots of components to what's the cause of this and how do you intervene in a way that's optimal for the baby. So, but what I'm saying is that for some of these infants that have the structural damage before preterm birth, it's not a typical nervous system that's receiving the dexamethasone, if that's what's being given to delay preterm birth.
So, yeah, so, I mean, so you're saying that, well, dependent upon the reason for preterm labor, preterm birth
Right, so there's
happening.
lots of
Yes.
potential causes, but a new one that's been identified because of the increasing use of fetal MRI. There's something that wasn't on the horizon,
Wasn't anybody's radar.
wasn't on
Yeah.
anybody's radar. So it's not maternal infection, it's not maternal stress so much as.
That's it. Yeah, that's interesting. So knowing that, once they know what the differences are in the fetal brain that cause preterm labor or whatnot, I mean, it would be interesting to see if their responses to Dex are different depending upon the brain structure. And it does depend on the,
Correct, that's what I was going for.
yeah, and
Correct.
it does depend on the time period as well,
So it doesn't
because
mean you have to have this structural difference before birth. But now you have a separate population of babies
yeah.
that have been identified as having. So anyway, that's.
Yeah, and that's, I mean, yeah, and that's also interesting because we know that GR expression changes throughout the brain before birth. So depending on what time period that is and what brain structure is
I'm not
underdeveloped.
sure when these fetal MRIs are being done, but
Yeah.
they're being done at children's field.
Okay, we have Lexi's doing fetal MRI. She did fetal MRIs in mice, rats,
Yeah, but I think
cerebellum, but
the point you raised, which is a really cool one, is to say whether or not it's legitimate to go back and do retrospective.
oh
studies.
yeah.
You know, looking back at those MRIs and seeing if those kids were given steroids or not.
And then see what the outcomes are. Are the outcomes worse if you've got a combination of glucocorticoids and evidence of structural differences compared to babies who didn't have the structural differences who were given glucocorticoids and then had a better outcome?
Or is that why some kids in some studies, they say maybe it contributes to cerebral palsy and motor deficits, while in other kids, it's just
So why the
ADHD,
variability?
you know? Yeah,
Why the variability?
yes,
And can you now start predicting greater risk factors
yeah.
for worse outcomes? Because you now have this piece of information.
There's your next, like he's writing down. There's his next med student project.
It's like shocking when you say that they're doing this for, like, yeah, they're doing it at children's, they're doing it everywhere. It's new technology. Feeding them across.
Wow.
Hey, we're gonna do this for our study and you don't have to pay for it. We're gonna get just like ultrasound, it's just you get better pictures, you know? The other question I had was about other SOX family members.
Yes. So, I have a few slides on this, but I will just summarize. So the question was about other SOX family members. So the reason that we chose to take SOX2 and run with it, one was because we had formed this collaboration with people that already had a transgenic SOX2 knockdown mouse. So one was resources. So that's why we ran with SOX2. There are studies that show, I'm not sure which three it is. It's like SOX2, SOX3, and SOX11 or something. They will bind to different enhancers in neural progenitor cells. And then as the cells progress and towards astrocytic or neuronal populations where SOX2 binds would change, which means that if it changes the GR responsive genes, that's why at different time points throughout the brain, if you would treat at earlier time points when it's more neurogenesis and not gliogenesis, maybe you wouldn't see an effect on glia. Whereas if you treat later on, it would be more on glia because those SOX2 or other SOX factors would be at different enhancers. So we focused on SOX2, but it would still be interesting to see, does GR bind SOX11, which has a different role than SOX2 in determining, you know, neural outcomes and things like that. So there are studies, a lot of studies, and someone else asked a question about SOX transcription factors, and they have their own cofactors that they associate, which can either be more stem cell-like or more neurogenic, depending on the time period. So there are a lot of avenues, you know, to go down with that. So we focus on SOX2 because mainly resources and the knowledge that it mediates chromatin looping. So I haven't seen any other chromatin looping studies with other SOX factors, but I'm sure they're in the works.
I don't have a ton of questions, but I was interested in the SOX mainly from the perspective of, I guess, the way that you pitched it, which is always, you know, gets my attention, which is that you didn't expect it.
Yeah, that's the one thing that we were like, well, there it is. It's relevant to ourselves, but we had no idea. We didn't know if we would have said what would we expect. We would maybe guess other proteins that are expressed in our cell type that are always shown in other cells. So like AP,
That's what I wanted to know, like, because you were saying that you thought maybe it was specific to your cell.
Yes. Yeah. So it doesn't show it. Glucocorticoid.
I like really want to break it down, if you don't mind.
Yeah.
Because I just wanted to understand. But so one is, what exactly do you mean? How do you define cell type? Is that like, are you talking about a homogeneous cell type?
Yes.
Stage.
So when I say cell type, I'm referring to our neural summer progenitor cell cultures, which are mixed progenitor cells. So they could be radioglial cells versus neural progenitor cells, which are a little bit more immature. It doesn't really include immature neurons because we keep them in this proliferative state. So proliferative stem cells, they could be radioglial cells. They could be neural progenitor cells. They're Nestin positive, SOX2 positive. So all the classic stem cell markers, but it's not it is a mixed proliferative cell type culture. So when I say specific to our cell type, I'm saying that in comparison to other GR studies, say in A549 cells, or basically yes, basically there's not a lot of GR studies in primary cell lines. So a lot of GR and how it works has been worked out in, you know, immortalized cell lines, non-neuronal cell lines for sure. So when I say specific to our cell type, I mean GR action in our cell type has this cofactor, as opposed to GR action in other cell types, this cofactor has never showed up. So that's what I mean by cell type.
And then and then I just wanted to, like, it just started at that bottom, but, and then, so what were you expecting? What did other people see and what does it imply?
So a lot of what we saw is not new. So we know it was not a surprise when we got the chromatin accessibility data back. Did you guys hear the question? Did you hear the question? Okay.
It's
So
all probably basic for them. This is not.
no, it's okay. A lot of what we saw was not new in terms of what did we expect to see. So the chromatin accessibility data, that GR does not change much of the chromatin landscape. That has been shown in several other cell types, so we just didn't, we had to do it ourselves in our primary cell line. The fact that GR binds to open chromatin as opposed to it doesn't, and it binds to a lot of distal enhancer regions, that was not new. So that wasn't necessarily a surprise. So the surprise was when we got to the cofactors,
Right.
essentially.
So that's what I mean.
Yeah.
What did you expect in terms of like what it might interact with, or what other people had seen, and why SOX, would be found as a cofactor
So
in your cells? Like, what is known about SOX that makes it make sense? I guess.
yeah, so where the connection is we know that both GR binding to specific regions of the genome change the proliferative capacity and the differentiation potential or trajectory of cells. And we know that SOX factors play a big role in maintaining neural stem cells or pushing them towards a neuronal lineage. So that's the overlap. A lot of that occurs in non-coding regions, which kind of makes it hard to say, okay, if it's binding to this place that's kind of in the middle of nowhere in the genome, where is the connection with it influencing cell fate? But I guess did we have expectations of what we expected to see? I didn't. Besides a GRE as our first one. So studies of cofactors in neural stem cells, there are some, but there aren't many. And in terms of glucocorticoid action in neural stem cells, there's not many other groups that do that. So it was kind of going off into uncharted territory a little bit. So some of it was exploratory. But once we found that SOX cofactor, then it became very targeted. Yeah.
So when you're surveying like the genome for these GRE binding sites, and then you look around them to see what other sites are there that resemble what transcription factor sites, right? I mean, are we talking about a needle in a haystack in terms of the ones that are accessible and actually interacting?
So like what percentage of GREs are bound at a certain time?
Yeah,
or?
like, are near the SOX, like in the genome.
I mean, it wasn't, I mean, before our study, we didn't know. If you're saying what percentage of the genome, I mean, it's a very small percentage of the genome overall. And an even smaller percentage of binding, near target genes.
I mean, these GRE sites.
I'm not sure that I'm understanding your question.
I'm not sure I'm understanding my question either. I guess I'm just thinking about it from the perspective of like how, historically, like people were studying gene regulation by just bashing at sites and trying to recognize sequences. And now you have these, awesome techniques. We have these techniques where you can look at what the open chromatin genome-wide study, and you can do the ChIP-seq. That's one's been around for a while.
Yeah.
And then you can correlate that or try to with transcription. But, if you're just looking at the genome itself without having all these peaks, can you look in with the rear view mirror and say, oh, like.
Okay.
Or is it a needle in a haystack? That's my question.
So only, yes. No.
Because I'm also curious about cell types in the open chromatin.
Yes. So just because there's a GRE at a specific spot in the genome doesn't mean it's going to be bound. So if we looked at the whole genome and said there's GREs here, 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 and here, that doesn't mean that we would automatically predict that those regions would be bound, because there's many other factors that one, guide the receptor to that specific spot and determine how long it stays there. The chromatin accessibility, you have to take that into account as well. So no, we didn't look at the whole, that wasn't an approach we took where we looked at the whole genome and said, we think GR is going to bind at these 1,000 sites, because there's so many regulatory factors that determine GR binding to specific sites and how long it stays on the genome, et cetera, that it's not trivial to predict.
What about the soft sites next to them? Are they, how close are they?
So we did
You
an
said
analysis.
it was like within,
Yeah, within 100 nucleotide base pairs is how close they were. They were more likely to be within 100 nucleotide
so if
base
you like
pairs.
looked and scanned the genome, like.
The entire genome.
The entire genome. We
Well, I
didn't,
mean, not
yeah.
by- Not by hand.
We didn't, yeah, we didn't look across, yeah. Yeah, so we only looked at those two motifs at target sites. We didn't look across the
It's like
genome.
1,000,
Yeah.
10,000.
Our bioinformatics team would be like, I don't know about this one, guys. So yeah, we didn't look across the entire genome with those two sites.
Good question.
Yeah, I understand. Yeah, you're just trying to visualize where everything
Look at the
is.
entire genome and say, what's close to it, the GR binding site over the.
Well, I think
entire
you
genome.
finally, like, I don't know, what do I know, but, you know, I think it's really interesting to break the code, if we can finally break the code of, you know, transcription, like, of gene regulation, and
Yeah,
if you know what transcription factors are present in your cell type, whatever you, however you define a cell type, and then you know, like, the open chromatin regions.
yeah. Well, the
And then
most,
you can, you know, and whether there's like gonna be new principles that can
Mhm.
come out of these kind of analyses,
So-
but then you can start making predictions.
Yeah.
Without necessarily having.
so the most important thing that you need for predictions is chromatin looping data. So it doesn't matter if there's something bound there. What matters is that chromatin looping that connects it to an actual gene, which gives you an actual biological response that's meaningful. So that's the most important thing, is the looping. So maybe if we had the chromatin looping data and we knew where these sites were and knew if dexamethasone changed the loops, then we could make predictions. But without that, then it would be really hard to make predictions based on the entire genome. That's the most important thing. So then that's why I mentioned in the talk, that's like the logical continuation of this that would allow us to identify some of these non-coding regions that are involved in disease.
That's where the field is going.
What?
That's where the field is going. It's looking, and that's.
And the GWAS Association
Mhm,
studies.
yeah.
To this, to the loop.
Yeah.
It's super expensive. You know,
Anyway, it's a nice, very nice job.
Thank you.
Okay, are we ready for round two?
So, I think we're done.
So I have a few more questions, Kim, and this just relates to, and I think if anyone else has anything that came up in the initial discussion, or any other ideas that they wanted to float by you, we still have a little bit more time. So my question pertained to things that you either didn't show or didn't do. And one of them is maybe a little bit with relation to what Mary was asking about the N of 1. So one of the reasons I understand that these things take a huge amount of time and energy and resources to complete them, but one of the other things that you could do is an approach that doesn't require all of that commitment of resources to validate or confirm the conclusions you make from that N of 1. And so I was just sort of wondering if there are things that you could do or did try or could have done to, you know, look at 5 mice, but only one site or something like that, like to confirm that what you're seeing from a really comprehensive screen from one animal actually, you know, maybe a few sites, and just kind of validate it. Are there ways that you could do that?
So right now, previous to ATAC-seq technology, the main method that people use to look at accessibility was DNA hypersensitivity assay, DHS. And those also are genome wide. In terms of looking at targeted regions of accessible chromatin, I don't know of any technologies that are commonly used or exist at this point. So so in terms of looking at individual sites of accessible chromatin, unless if there's something I'm not thinking of, which I know Dr. Singh would be an expert on this point, there's not a way to look at a targeted region for accessible chromatin.
Old school. There is. It's very old school.
Oh, that says there is a way that's very old school. So no, but we did not try to do that.
And then I guess maybe
Sometimes people do chip qPCR against specific marks, but that's about it.
Like histone modification markers.
Yeah, but like you design your primers against a specific region. So instead of after immunoprecipitation, instead of going for full sequencing, you just quantify how much protein, whatever, if you're going for a transcription factor or a histone mark was present in that locus.
Okay, so you're saying you could look at histone markers that would say this is an enhancer, so it's most likely open and active, but it doesn't directly tell you about that it's open.
No, what I'm saying is like you can design a chip. So you can, if you have identified a region which you know is maybe accessible or is a region of interest by ENCODE data or something, like, because with ENCODE they have profiled so many cell lines, so you kind of know that these regions are potential enhancer elements. So you can design primers against those regions and quantify how does accessibility change based on your treatment. So this will be like a small scale, not going for sequencing, but still quantifying the accessibility. They're selecting a K27 acetyl kind of marker.
Okay. Yes.
PCR rather than sequencing.
Yeah.
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 And doing that sort of thing, Kim, do you think it would be better to pool 5 mice or individually assay them, or what would you think would be the most desirable way to design something like that?
I think it would be more desirable to do them individually first and look at biological variability between samples before you pull them together. Because if you pull them together, maybe you might lose some I would do it individually first before you pulled them together to look at the biological variability. I know that the chromatin landscape changes depending on the cell type, and even if you do single cell chromatin accessibility, it's different between individual cells. So I think there's a lot of opportunity for variability to be introduced if you combine embryos together.
Okay, and then the last sort of question about what you didn't do or didn't show is an alternative to the PLA. Is there any other way that,
Yes.
that?
So we tried several things. We tried doing co-immunoprecipitations first. So trying to pull down GR and SOX2 together and look at protein analysis. That was unsuccessful, just method wise. I tried for a while I couldn't get the IPs to be very consistent. That's the biggest thing that we tried with before the proximity ligation assay to look at protein proximity was doing immunoprecipitations with large amounts of protein. And a lot of times, so we would first do an IP for GR, and then I would take that protein lysate and stain for SOX2, and I just couldn't get it to work consistently. So the proximity ligation assay is kind of the second thing that we tried, and it ended up working. The proximity ligation assay I also tried to do in tissue, but it's just technically tricky. And it wasn't just in my hands, in other hands, it was technically tricky as well. So the PLA assay in cells is the only thing that I was able to consistently get to give us some reliable data.
Especially given that you're using cells, is there any other approach that you could think of to use in cell culture aside from relying on antibodies?
We also tried to do RNA interference assays. So we also tried to maybe knock down SOX2 and see if that affected the DEX effect on gene expression just by doing qPCRs on target genes. So that's another way that we tried to look at that interaction, and that doesn't involve protein. But that again was tricky. These cells are very finicky. So if you introduce a construct or if you do electroporation or something very mechanically abrasive, the cells don't react well, they die quickly. So we did try that as well to look at the interaction, but again, you know how experiments go in the lab, it didn't pan out.
Okay, that's all the questions I have. If anyone else has any other questions, this would be the time. Okay, if no one else has anything else for Kim, I guess it's time for you to step outside of the room, and the rest of us will confer.
All right.
Is
Patty
this-
already sent the DocuSign.
I know, I think we can dispense with the formalities.
Don't go too far.
Okay, I'll stop right here.
Okay.
I think she did a horrible job and she should stay for long.
And replicate herself like 3 or 4 times.
What is she?
So,
doing? Or
so I think just to say the main point of this conversation, I think, is just to give Kim some feedback when she comes back into the room. I mean, my personal impression was that she's fabulous and did a fabulous job. The document read like better than most scientific papers I've ever read. So, you know, I think she's, I only have very small amounts of feedback for her that would be helpful. And I'm happy to send it to her separately. I don't have anything really critical to say. Does anyone else have anything that would be helpful for Kim?
Yeah, I think it'd be helpful to give her feedback about her writing and her analysis. I mean, that's her strength. Her strength is, you know, her communication skills,
Yeah.
her writing.
She's always been top notch.
You know, I mean, in the lab, I think some techniques were just really hard for her. She spent months trying to get QIP to work. She spent months trying to do the RNAi knockdown. But, you know, and so technically, I think as a hands-on bench person, and that's not what she wants to do in the future, because she's looking for other jobs. But she recognizes her strengths, and the fact that she dove into this informatics, you know, and she really helped direct this. I mean, she didn't do the informatics herself. We had Uma and Feng Ping, but she directed them and she analyzed the data together, and she said, why don't we try this? And it was very impressive to see how much she really learned about informatics, you know, from someone who was not trained in that area. So I mean, I think she would really appreciate the feedback that she's become a really good critical thinker and writes well, communicates well, and to keep pushing that forward as she should move forward in her career. But, like, you know, I want to thank all of you, because I know it seems like it's been short, but it's been a long time. And certainly too for our guest, Tisha, thanks for joining us in. You know, I think any feedback you have on some of the things we could do with informatics would be very helpful, because we can basically rely on Uma and Feng Ping to look at data sets that are out there. And I know your work in GBM could be, I mean, this could be very helpful for us too, that we didn't require any more lab work, but we could make these directions. So if you have any other feedback for informatics stuff that you think would be useful to move this forward, I'd be really grateful. But
Yeah, I think with the amount of data she had, I think she covered most of the analysis that you can cover with that amount of data. So I think I'm satisfied on that front.
okay. So Sean, what do you want to do?
Well, I think she should pass.
Okay, should we bring her back in?
Yeah. And Don, do you want to give her the good news, since I'm on Zoom? It'd be a little nicer.
I'll wait till I bring her back in here. So, so I think I'll be the one to say that it's unanimous, congratulations, Dr. Berry. Everybody was very impressed with, certainly the thesis writing was, and Sean can reiterate that, found it really super well done, critical thinking, the whole progression, communication skills. These are all big strengths that I think are gonna really help you as you move forward. And if anyone else wants to add anything.
Yeah, I would just say, congratulations. It's a huge amount of work. And I mean, I personally really enjoyed the update on the latest in the field of glucocorticoid research. I mean, this was the subject of my doctoral research. So I kind of tuned out for the last decade, and to get to tune back in through your lens of critical, I mean, it was wonderful to see things that were like old friends come back, where you talked about the literature that I had reviewed, and when I was doing, in your shoes, doing what you were doing, and then to see the latest updates was so incredibly exciting and just so clearly articulated. So I mean, and I'm happy to give you some feedback about it offline later.
Yeah.
If you'd like to chat a bit more about specifics, areas to continue to develop and work on, we should find a time to chat before you leave. But super impressed. Really, really awesome job.
Thank you. I appreciate it.
What are you gonna do next?
So I'm looking to go into medical communications, like science writing jobs at the moment. I'm in a couple interview processes right now, so nothing for sure, but that's where I'm at. So
What kind of medical writing do you want to do? Or what are you?
at first, whatever I can get. Eventually I want to go into, have you guys heard of a medical science liaison? Yeah, so eventually I want to go into that, but you need to get your foot in the door into, industry in general. So I plan on doing science writing first, and then transitioning to medical science liaison when I can. So that's my goal. That's my trajectory. So
Nice. So yeah, I want to reiterate, excellent job. Your presentation was awesome.
Thank
Your
you.
document read so well, and it's a lot of stuff that I'm not very strong in terms of genomics and that kind of stuff. And I understood everything. So
Oh,
you did a great job.
great. Thank you. That makes me feel so good. And I know you have a couple students that they have this coming up as
Depending
well,
very
right?
soon,
Yes.
yeah.
Yes. So thank you. I appreciate it. Thank you. That makes me feel good.
Yeah, certainly, you're very strong in those areas.
You made it.
But I'm just impressed with the way that you pulled all of this stuff together,
Uh-huh.
It's a piece of story that's got a lot of like dangling, interesting bits for people to go on.
Yeah.
Like, I mean, impactful.
Well, Don is the expert at seeing connections and seeing the next directions. Like, oh, we could do this, we could do this, we could do this, we could do this. So I think you trained me well in like thinking of all the implications and different ways that things could, bend. So
Like I said, save the best for last.
yeah.
Yeah, it's a lot of data that you collected and analyzed.
Oh yeah, we were in the weeds for a while. Yeah.
So formally, what do we do, Sean? There's certain things we need to sign.
I think it's all done.
Yeah. So
Yeah,
Patty circulated the DocuSign, I think, right? Everyone got that?
I signed it also.
Okay.
Then we'll have to submit the final version.
Yes. So for submitting the final version of the thesis, is it mostly good as is? Are there any like major?
Oh yeah. So we would have told you if it needed rewrites. That was yeah. You're it's great.
Amazing.
Don't know how many DocuSigns we have to sign. 'Cause we were signing
There might be two different
two
things.
different things.
Yeah,
So.
Yeah.
just make sure you sign everything.
Well, good luck.
Thank you. Thank you guys
Yeah.
so much. I really appreciate it.
Thanks, thank you, everybody.
I've loved
Great.
having a committee of mostly women. I'm sorry, Don, but but I really have loved it. It's been amazing. So thank you guys so much.
Okay,
Now,
well,
I know, like, what do I do now? I don't even what do I do with myself.
don't thank your father and show him.
I know.
Don, did the senior retire?
What's your?
24.
Okay. Thank you so much, Dr. Singh, is she still on here?
About-
Yeah, she is. Thank
Okay.
you. Yeah.
Yeah, thank you so much for tuning in. I hope this experience has been a good one.
Yeah, thank you for having me.
Thank you so much. I'll keep in touch.
Yeah.
Okay, and Dr. Sorrells, yes, I'm definitely would love to have that meeting, maybe next week, maybe not this week.
Whenever. Take some time. Yeah, no hurry.
Okay,
Yeah, congratulations.
I thank you so much. I appreciate it. And thank you for being, I'm glad that you were the head of my committee. You made it really not informal, but very friendly and open, and I appreciate like your approach to how you did it. So is this your first one?
Maybe I can't keep track. I think maybe so. Yeah, maybe there was one other where I got roped in last minute. I can't remember.
Okay.
But yeah,
This is the first one as like the head of the committee.
yeah.
Okay, well, I really enjoyed it.
Thanks so much, Sean.
Me too. Yeah, no, thanks so much, Don. Sorry I couldn't be there in person.
That's okay. All right. Thanks.
Congratulations. All right, have a nice day. Bye-bye.
All right, that's it. Congratulations.
Oh, Don, give me a hug.
You did great.
Thank you.
It's fabulous.
Wow, I didn't cry today.
I almost did.
Oh my gosh. I cried yesterday because I was like, oh, I was just having anxiety.
It is, I mean, it's weird for me. I'm gonna be honest with you. Because it really is like you're my last student.
Yeah, you're done.
Yeah.
You still got a few more years.
I mean, yeah, I know I'm sitting around, but it's just,
All right, how do I start this?
it's awesome. So now, yeah, so I guess